Hello everyone, am I clearly audible to you all? Okay. Let us start about studying the instrumentation. Now, before starting instrumentation, I like to tell you that this particular lecture I am hello, good afternoon. Uh, Good afternoon, Toxic Gamer. I don't know what's your name. So, this particular lecture is actually made on the request of my few students. The one is Fatima and all. So, they just wanted to know about the instrumentation deeply. So, that's why we have started this particular instrumentation lecture. So, the instrument suggested by my students only will be discussed into this particular lecture. And next time whenever we will have time we'll discuss the other instruments. So can anyone please tell me into the chat box that am I clearly audible to this session? Come on, fast. Okay, now see students, when I talk about the instrumentation, so instrumentation is a study about the instrument. What is the relation between the zoology and instruments? Is there any relation? Is there any relation between zoology and instruments? Yes, it's absolutely there. Why so? Because the just a minute, let me check the why the instruments are so important. Now see, the instrument help us to find out the various type of concentration. It help us to see when I talk about the microscope. So definitely instrument help me to see the biological samples in a broader view. If I talk about the separation, so first thing we will start about the separation. If I want to separate the substances, there are lot many methods. From six standards we are learning, we can separate the method by these, by that, right? So how can we separate the substances? We can separate, अगर solid to solid है तो we can simply separate it by the hand picking. Daily का जो चावल आप clean करते हो, we can separate it by winnowing, sieving. We can separate it by threshing and lot many things. If you want to separate the solid to liquid, you can make use of the strainer. You can make help of the filtration. Liquid to liquid, if you want to separate, you can take help of the distiller. Now, if you want to separate some more biological substances, you can do the chromatography. But now you tell me, how will you separate this sample? How will you separate? If you want to separate the components of the blood, how will you separate? I want that component of the blood to be separated. Plasma alag chahiye, red blood cells, then apart from that white blood cells, everything I want to separate. How will you separate this thing? Think. Now, for separation, what you need to do is that you want that the heavy materials like the red blood cells and other components which are heavier. Yes, Huda, we can separate it by sedimentation, but which machine we can use? How can we separate it? So we can separate it by the process of centrifugation. Now see, centrifuge machine, if I talk about, so the first centrifuge machine was made to separate the cream, not the blood sample or nothing. So the credit of making first official centrifuge machine goes to the Gustav the level. The he made this particular centrifuge machine in the year 1894, and for the purpose for separation of such machine, for the separation, very good Fatima, very good Fatima Sheikh. It's a centrifuge machine, and Huda also. So centrifuge machine. is made by the Gustav the level in the year 1894. Now, see, he didn't make this particular machine for the separation of the biological samples like blood. We saw his idea and we did modified it in for the separation of the clinical material. He made it for the separation of the cream. Now, see, basically, if you will go to the Rajasthan, do you remember in regular lecture we were discussing about the Rajasthani people? They are making the makhan, so they are making use of this. Well, they are having the churning bowl in that they just churn this milk, and after coagulation they are separating it. So same way, but that is such a tedious process. If you want to go it on the economical level, definitely you need some machine. So he just devised this particular machine, and say when we are talking about the centrifuge machine, so as I told you earlier also that whenever we are talking about any machine, so what we will remember its principle, then its construction, then its application. So these three things we need to remember. Now see, centrifuge machine is modified to different type of centrifuge machines, so we will be discussing it later. So first we talk about the principle. Now what is the principle of the centrifugation? So the centrifugal force can be used to separate lighter particle from the heavier portion in the liquid medium. Lighter particle from the heavier portion. So this is important to remember that centrifugal force, actually centrifuge machine works on a centrifugal force. What is the centrifugal force? A force which is applied from the center, and from the center it is passing. So this is called as the centrifugal force. So a centrifugal force is used. Why the centrifugal force is used? It is used to separate the lighter particle from the heavier portion, and this can be done only into the liquid medium. The centrifugal force begins greater than the gravitational force, so that's why it can separate the portion of different densities. Now there is a simple equation which runs behind this particular force that we need to remember, because this is a particular type of equation is important marks point of view, और ऐसे भी हमें याद ही रखना है. So the equation is F is equals to m omega square r. चलो देखते हैं ये क्या है. F is your centrifugal force which is applied by your machine, and m is the mass. What is the mass? Mass is the material which is present, or substances which is present into the liquid जिसको आप separate करना चाहते हैं. So m is the mass. What is the W here? This is an angular velocity. W is not actually omega. It's not W. I'm sorry. Omega is the angular velocity of the particle, because here we are, when you will be applying the centrifugal force, so that particle is going to rotate. So that rotation will have an angular velocity. So this angular velocity is called as the omega, and r is the radius from the center of the circle. तो यहाँ पर दो बातें बहुत इंपॉर्टेंट है याद रखने के पॉइंट से. द फर्स्ट थिंग इज द RPM, revolution per minute. That means after applying the centrifugal force, how many revolution this machine is taking in a minute. I'm sorry to interrupt. Fatima, ask everyone to join immediately, because I can see only 5 of you have joined. Ask everyone, because I am not going to repeat again and again the things. I'm so sorry. Okay, so revolution, when you are applying the centrifugal force, so in every minute, how many times that angular velocity is happening? So rotation per minute. And the next important thing is RCF. That what is this RCF? Relative centrifugal force. Now see, not only revolution per minute is important, but also the RCF is important. Why this RCF is important? Because see how much force you are applying, on the basis of that particular force your revolution per minute is related. So RCF and RPM are related. So what we need to remember that the force acting on the sample is depend not only on the speed of rotor. Now this is also depend on the radius. How many big radius are there? Suppose if you are spinning two different sample into the two different rotor, both the rotor have the same speed, but you have taken smaller radii in one of the centrifuge machine, and in one you are taking the big normal one. So where the force will be more, the force acting will be more into the smaller radius because the smaller area will be affected. However, if equal RCF values are applied to sedimentation forces will be identical. Same hai. As such, RPM value will be informative only when the rotor type or radius is specified. So centrifuge, why do we use centrifuge? It's very essential instrument in cell and molecular biology, because until and unless you will not separate the cell, so you cannot study them. It is generally used to separate the biological component based upon their differential sinking properties. So that's why we are now see, if you will see the biological components, so their SD value, that means sedimentation value is noted. जैसे कि आप लोग इसका देखोगे राइबोसोम्स का, द राइबोसोम्स वैल्यूज आर मेंशन इनटू द 70 है 60 है. व्हाई सो इसका कोई प्रॉपर मास वैल्यू क्यों नहीं लिखा होता है? बिकॉज at how much forces they are sedimenting, they are important on the basis of their unit has been mentioned. So what is the construction of the centrifugation machine? That first you have a suspension over it, and on this particular suspension you are applying the force in, and that force is in process, and induced gravitational field will be happening, and point to be noted at this particular that the centrifugal force is more than the gravitational force. And after that, applying the forces, the lighter particle will be become the supernatant, and whatever is sedimenting is going to be called as a precipitate. Now, send on the basis of the RPM value, that means revolution per minute value, the centrifuge machines can be divided into the various types. Lot many ultra centrifugation, low speed centrifugation, high speed centrifugation. Lot many type of centrifuge machines are there, but we will stick to only and only two type of centrifuge machine. One is the low speed centrifuge, and next is the high speed or the ultra centrifuges. Now when we talk about the low speed centrifuge, you need to remember the low speed centrifuge is also called as the clinical centrifuge machine. Low speed centrifuge machine is also called as clinical centrifuge machine. Now where do we use this low speed or the clinical centrifuge machine? The this particular machines can be used to use into the laboratory. That's why it is also called as a clinical centrifuge machine. And we need to remember that it is found only into the biology laboratories, or maybe you can also see it into the chemistry laboratories to separate the sample. Now revolution of this machine is 1000 rpm, that means 1000 revolutions per minute. But you will see that slight modification, as we can see in our laboratory. We have 1 2 3 है ना, the setup, that button is there, in that we can set it up to the 3000 revolution per minute. So maximum 3000 revolution per minute can be, sorry, 30,000 revolution per minute can be seen. It is a mistake, it is not 1000 rpm, it is the 10,000 rpm. So up to 30,000 revolution we can see the normal machine which we are using in our laboratory. So it is having up to 30,000 revolution per minute. So I'm very sorry, students. So let's talk about its application. So the application of clinical centrifuge machine is that we can use it into to for the separation of the biological samples, like serum or plasma, we want to separate from red blood cells, and to harvest the purified chemical precipitates we want intact cells. Then we want nuclei, larger mitochondria, or maybe the large plasma membrane fragments, etc. Usually, basically, this particular centrifuge machine is used for the separation of the plasma and serum. When you will go into the TY, that time you will learn that lot many practicals we are doing on the plasma, because this plasma is having lot many antibodies and other relevant substances which can be used to find out the protein estimation, then your lipid estimation, lipid profile. So lot many things we need to check, or you can say the hematological assays are done on the plasma or maybe the serum. So for that separation of the plasma and serum, we are using the low speed centrifugation machine. Now next we will talk about the high speed centrifugation machine, that is called as the ultra centrifuge machine. Now see, this ultra centrifuge machine can achieve rotor speed over the 30,000 rpm, और उससे भी ज़्यादा ही होता है, यहाँ पर भी 33,000 हो गया है एक्चुअली. I type it late night. So we can see that we can achieve the over the 30,000 rpm, because I told you up to 30,000 rpm we can set up into our low speed or the clinical centrifuge machine. So यहाँ पर above that only values will be there. Now see, when a lot of speed is been applied, so definitely lots of centrifugal force we need to apply. So that's why we need to use the heavy motors, and when such heavy motors are being used, it is going to generate a lot of heat. So for that particular reason, we need to create a high quality refrigeration system where the biological sample will not be heated, because when the protein is being heated, so because of that particular heat, the protein gets denatured, and lots of properties been degenerate. So for that reason, if you want to harvest the live cell, definitely we need that the system should be very clean. So for sorry, very cold, so that the harm to the cell will not be happen. So for that particular reason, we can make use of the refrigeration. And apart from the refrigeration, you can see the high quality vacuum is there, and just a minute. So a high quality vacuum is also been generated in this particular type of the centrifugation machine. They are used now. Where do we use? So see, such more complicated system. Say example, you want to separate the cellular components, like you want to separate Golgi apparatus, endoplasmic reticulum, endosome, ribosome, DNA, RNA, such complicated things you want to separate. So, in spite of doing a big protocol, now you are no, you don't have in your syllabus. SYBSC is having syllabus, DNA and RNA separation. So, in spite of creating such a big particular protocol following, you can just do it centrifugation. So, with the help of the centrifugal force, the biological sample not only sedimented but also it has been separated. So this is all about the centrifugation. If anyone have any doubt, please write into the comment box. Don't write into the comment box, write into the live chat that is it okay to continue with the next machine, or you want to know more about it, or you want a revision of the centrifugation machine. Come on, fast. Right now you post the comment into the live chat. If you want me to revise the machine once again, we will revise it. And if you want me to go ahead, we will go ahead. I'm waiting for your comment. Yes, Huda, ultracentrifuge is same as the refrigerate microcentrifuge. Actually, refrigerate microcentrifuge is having the microtubes that is called as the cryotubes or the cryo preserving materials we are using to avoid the lots of pressure in that. And refrigerated centrifuge is that. Okay, Fatima will revise the centrifuge machine, but this revision will be very quick. So we learned that the first clinical centrifuge machine was made for the separation purpose for, and Gustav the Level grants the credit of creating the first centrifuge machine, and he made this particular machine for the separation of the cream, and he made this particular machine in the year 1894. So the principle of the centrifugation is the centrifugal force can be used to separate the lighter particle from the heavier portion in a liquid medium. I repeat, the principle of the centrifugation is centrifugal force can be used to separate the lighter particle from the heavier portion in the liquid medium. The centrifugal force being greater than the gravitational force can separate portion of differing densities. It can be given as F is equals to omega square R. F is the centrifugal force, m is the mass, omega is the angular velocity of the particle, and r is the radius. Now we need to remember two most important parameters. The first parameter is the revolution per minute. So revolution per minute, that means when you apply the centrifugal force in 1 minute, how many revolution यानी कि कितने राउंड जो है आपका centrifuge आपका मटेरियल लेता है. Okay, that rotor is taking how many revolutions. So revolution per minute is what? One motor is taking how many round in 1 minute. So it can be explained it. Just a minute, let me take the screen. Why it's not coming by? I'm not getting the pen. So say example, if my centrifugal force is started from here, so till here, so this is one revolution. So likewise, how many revolution in 1 minute the centrifugal motor is taking? So this is called as the revolution per minute. Okay, so what is the revolution per minute in 1 minute? That how many revolutions your motor is taking, that is called as the revolution per minute. And what is the RCF? RCF is the relative centrifugal force. That means in 1 minute, how much force is applied to the analyte to take the revolution. So that is called as the relative centrifugal force. I hope this is clear what is the RCF and RPM. So if a sample and what next thing is important, the next thing is the how much radius is there. So यहाँ पर three things are acting. The first thing is the RPM, second thing is the RCF, and third thing is the radius. So on the basis of the revolution per minute, how much centrifugal force is there, on the basis of that the particles will be separated. So we can say that the centrifugal force is a force which is applied on the suspension, and when it has been applied on a suspension, so on the basis of that particular force, the heavier particles will be separated from the lighter particles will be separated from the heavier particles into the liquid medium. So on the basis of the RPM value, the centrifuge force are divided as the low speed centrifuge which we are using in our laboratory, and this low speed centrifuge machine is having 10,000 rpm, and it can separate the serum or plasma, it can separate the cells, nuclei, mitochondria, and the large plasma membrane. Second type of centrifuge machine is the intracentrifuge machine or high speed centrifuge machine, or we can also call it as a centrifuge, refrigerated centrifuge machine. So it is having above 30,000 rpm value, and it is having the refrigeration and the vacuum system in it. And the next thing, we can use it for the isolation and purification of the membrane component of membrane like Golgi membrane, endosome, ribosome, DNA, and RNA. Now, next machine we are going to talk about the colorimeter. Any doubt in centrifuge machine? Let's go and talk about the colorimeter. This is also suggested by one of my students to explain the colorimeter. Now see, this total principle, or you can say the formation of the colorimeter, is based on the electromagnetic radiation. स्पेक्ट्रोफोटोमीटर में भी जो है इलेक्ट्रोमैग्नेटिक रेडिएशन ही हम लोग यूज करने वाले हैं. So whenever electromagnetic radiation is interacting with your analyte, so there are three types of fate. Either that particular light will be reflected back when it is coming. Any electromagnetic radiation, when it is coming on the matter, it may be reflected back. Okay, it may be absorbed completely. Completely absorb होगा तो क्या होगा? That it will not come back. और इट मे बी द पार्शियली एब्जॉर्ब. तो अब ये तीनों चीजें किस केस में होंगी? रिफ्लेक्टेड बैक, दैट मीन्स इट मे बी अ मिरर सरफेस. Okay, maybe it is a metal. So in the case of the mirror or metal, when electromagnetic radiation will be falling, it will be reflected back. ये हमारे काम का नहीं है. If it is absorbed completely, that means the matter is black color, or maybe this particular matter is an opaque system. So this is also no matter. But if it is partially absorbed, so some will be absorbed and some will be passing through. So if we know that how much light is been passed in a length and how much is reflecting back, so we can count how much analyte is present. So this partially absorbed light can be used to identify that how much amount of an analyte is present in a given sample. So how can we do, and what is the principle of this particular machine? So the principle of colorimeter is amount of an analyte in a given sample. It is when placed in a cell of known path. Here the most important thing that we should know how much path that light is going to follow. Okay, so when it is kept into a system or in a cell of known path length, it may be estimated by allowing this particular light to interact with electromagnetic radiation of certain fixed wavelength. Now, why certain fixed wavelength we will discuss later, because each and every wavelength light can absorb only a simple type of complementary color. Okay, that we will discuss into the next slide. So when you will be keeping it into the visible region and measuring the intensity of transmitted electromagnetic radiation by such sample, so whatever light is been absorbed, that much amount of sample is present. So that much amount of analyte is present into the sample. तो बेसिकली वी कैन से कि हम लोग कॉन्सेंट्रेशन को फाइंड आउट करने के लिए ये पर्टिकुलर इंस्ट्रूमेंट का यूज कर सकते हैं. So wavelength which are been used in this particular machine ranges from 300 to 800 nanometer, or this particular wavelength is also called as the visible range light. Okay, 300 to 800. So we are using the visible. Why visible light is called as? Because this particular light is visible to the human eye. Wavelength of light absorbed by the analyte is depend on the principle of the complementary color. So let's discuss about the complementary color. I'm so sorry. Now what is this complementary color? Say example, now see when you will be making the substances. So if you want to make use or you want to measure the concentration of the substances, that must be translucent. Matlab, it should neither be transparent, it should nor be the opaque. It should be translucent. That liquid should be translucent. Why? Because just now I told you that if the substance is translucent, so the light will be partially absorbed, और जो बाकी का light pass हो रहा है, that light can be used, or that can be count to find out that how much concentration of the analyte is present. So here what can we do is, say example, if you have the yellow green solution, students, I will suggest you to take the screenshot of this particular slide. This is important. Students go to the MSc level, still they don't know के आपको कौन सा पर्टिकुलर सब इसके ऊपर ओडी लेना है. Many time it happens that intentionally some examiner in ask in viva okay, your substance is so and so color, so you are going to measure it into the which nanometer, so they forget, they are not able to answer. So I just want hardly here seven colors are there, you need to remember the seven colors and their respective wavelength. That means if you have and believe me, I have given efforts to create this slide, so to give it colored so that you can remember. So I just want that everybody should take the screenshot of it and remember it by heart it. So if your substance is yellow green color, that means in the visible spectrum it is going to absorb the violet color light, and this violet color light is having the 400 nanometer wavelength into the visible. बच्चों ऐसे जब विजिबल लाइट आती है तो विजिबल लाइट इज नॉट लाइक कि वो सिर्फ 400 नैनोमीटर की आएंगी, 435. इट इज नॉट डेफिनेटली गोइंग टू कम टू दैट पर्टिकुलर वेवलेंथ. what we need to do is we need to place a filter, which filter I'm going to explain into the next slide. So this filter is going to choose only and only 400 nanometer light which is having the visible spectrum of violet color light. Now why I am using this? Because substance of my solution is yellow green, and if it is yellow green, so that means they are going to absorb the violet color light which is present at the 400 nanometer. This concept is very important and it's really unknown to many students. They go to the MSc level, they don't know okay, what nanometer they have to set. So I just suggest you to remember this particular thing. If your solution is of yellow color, it is going to absorb the blue color light, and you are going to use the 430 nanometer filter, 435. If your substance or your solution is purple color, it is going to absorb the green color light, you will be using the 495 filter. If your substance is blue color, it is going to absorb the yellow color light, and you are going to use the 560 nanometer. Remember, protein purification, at the time of protein purification you will be getting the blue substance. If your substance is greenish blue, so you are going to make use of 650 nanometer wavelength light, because it is going to absorb the orange color. Then if it is bluish green, it's going to absorb the red light, and you are going to use the 800 nanometer filter. Okay, so it's clear to everybody that what type of filter we are going to use. Now let's see how this filter looks like. This filter is something like this. Now this filter, if you will be selecting the wavelength, it is going to filter. That means this is going to allow only and only that particular light from the visible spectrum. Now how it happens? So for that we need to understand how this particular uh, construction of this instrument is. So there is, we are having one light source, and from this light source visible light is passing. So there is an aperture, or you can say there is a slit which is going to you can say align the lines into the parallel waves. कैसी भी light आ रही है ना कोई भी direction में. So this aperture is going to frame the light to pass in a straightforward direction so that they can propagate into the straight line manner. And after that there is going to be a condenser, or you can say there is going to be a lens. This lens is going to condense the light to passes on a filter. Now here in the filter we have seen that only and only selected nanometer wavelength light is going to be allowed or is going to be passed. Now this light will be passed on a cuvette. Cuvette, a special type of container, is used which is opaque from the two side and it is transparent at one side. Now in the cuvette special type of quartz materials are being used so that it will not allow the reflection of a light and it will not refract also. It will pass the light into the straight direction. So for that reason these cuvettes are made up of the very special quartz type of material, and in the cuvette your analyte or your sample is present, and in your sample whichever color matter is present, so how much light is been absorbed will be reflected, will be absorbed by the matter, and the remaining light will be passing to the photo visitors, or you can say simply photocell. When this photocell light will reach on the photocell, that will create a signal on the galvanometer, or you can say the machine which is going to show you detect how much light has been absorbed, and you will be getting the absorbance. So once again we will understand the construction of the colorimeter, that there will be a light source and the light source will be passing the light of the visible spectrum, and this light will be passes from the aperture slit and that will be condensed by a lens. From that it is going to allow to fall on a filter. That filter will be select the light of the selected nanometer wavelength will be passed on the analyte, and analyte will be absorbing the light of the selected spectrum, and the remaining light will be fall on the photocell, and that will give a signal to the galvanometer, and you will be able to find out that how much light is been absorbed. तो यहाँ पर एक बात याद रखेंगे, what if your sample is colorless? Say example you want to test a sample which is colorless, so what you will do, you will react with some type of the chemical. जैसे कि अगर if you want to say you want to measure the ammonia, okay, कितना ammonia present है आपके fish tank. So now ammonia is colorless, but the ammonia level, if it is increased into the fish tank, it may be toxic for your fishes. You want to just simply measure that how much ammonia is present. So what you will be doing, you will be adding the copper solution in that. So ammonia and copper solution will be react, and you will be getting the dark blue color solution. So intensity of that blue color is directly related to the ammonia present. So if you will be measuring that, if the absorbance is high, that means high level of ammonia is present. Okay, so same way you can do. Now this colorimeter is basically work on the Beer and Lambert law. What is this Beer and Lambert law? It is important if it has been asked, write the principle and application of the colorimeter. So it is important for you student, exam point of view I am talking about. It is really important to write about the Beer and Lambert law. So Lambert has studied the relation between the path length and intensity of incident and the transmitted radiation by an object of thickness. So what is this actually? Basically the Lambert, it has studied that how much thickness or how much intensity is present. So it is given by the IO divided by the IT. Actually this is I0, IA is nothing but the absorbance is equals to KB, where the K is something you can say K is here, we can say is the constant. Okay, so this constant, and the B is what? B is the thickness of the object. Same way, the beer has studied the relation between the concentration and intensity of the incident and the transmitted radiation by the sample of concentration, that how much concentration is present. So here also absorbance is related to the Kc. Now here again, K is nothing but your constant, but here we have taken the K dash, so that when we will be combining the two reaction, will not be just get confused कि ये K था ये K था. So for that reason I have written the K dash into the C, where the C is the concentration. When we will be combining the two reaction, we will be getting that IOIT is equals to KB into K dash C. So K को एक साथ किया, and BC together I have kept. So it is going to I have just told you that IOIT is nothing but the absorbance, how much absorbance is happening, and this is equals to K, and K dash I have combinedly written as the A. You can also write it as A dash. B is the thickness and C is the concentration. So this is the important, or you can say this is called as the absorptivity of the sample. So we can say the absorptivity of the sample is depend on the path length B and the concentration. Now where do we use, before that I just want to tell you that the cuvette is your path length. Okay, what is this path length? Don't get confused. Cuvette is deciding the path length, that how much amount is passing. Now where do we use this colorimeter? Applications are important. 5 marks के लिए पूछा जा सकता है. Just write down the application of the colorimeter, and if application construction will come together, so principle and application will be asked, it will be asked for the 10 marks. So where do we use the colorimeter? To determine the amount of analyte present into the sample. This is usually what we do. We make the substance react, we do the chemical reaction, and after that whatever color we develop, so on the basis of that we determine the amount. Then it is been used to, I'm sorry, it is also used to know the molar absorptivity. If you know the molecular weight of any analyte, so how much molar absorptivity of that sample is there, for that we can use. Then colorimeter also be used for performing the titration to determine the strength of given solution in term of gram per liter. So such type of titrations are actually called as the photometric titration, and these are possible if any of three essential components like titrant, solution of known concentration, titrand, that is the solution of unknown concentration, and product of titration absorb of light is given, wavelength is known. So that time we can use the colorimeter for knowing the titration. Now colorimetric analysis can be used for following the progress of any chemical reaction with the time. Say example, activity of certain enzyme like amylase or something you are using for the digestion of starch, it can be used colorimetry. So if the digestion is happening, so the solution will be becoming translucent. Same way you can also use it to find out the density of bacteria. So if you are diluting the bacteria, you will be doing it into the TY. What you will be doing, you will be incubating the bacteria, and after that the various dilution you will be taking into the flask, and with the help of that side-arm flask you will be measuring the activity, or you can say the amount of the bacteria present. So this is all about the colorimeter. Any doubt into the colorimeter you want me to revise, or we can stop this note? And if you have any doubt, write immediately. If you want me to explain this again, kindly immediately write, or we can stop at this note. Okay, so I don't think you have any doubt. If you would have asked me. So we can stop at this note. I wish you all the best for your exam. So that's it from my side. Okay, we'll just take a quick review of this colorimeter. So we have seen that the colorimeter can is used to find out the amount of analyte, and the principle of colorimeter is based on the Beer and Lambert law. So how much analyte is present, that is going to find out with the help of electromagnetic radiation, which when passes from the fixed length into the visible region and measuring the intensity. So this visible ranges from the 300 to actually 800 nanometer, but in your book also 700 is given, but visible range is 300 to 700, but in colorimeter we can select 300 to 800. The light absorbed depends on the complementary color. What are those complementary color? The complementary colors are the yellow, green color का complementary color है violet, which is present at the 400 nanometer. Yellow का complementary color is blue, and that is present at 435. Same way purple का green 495 nanometer. Blue का yellow है 560 nanometer. When I will be checking your paper, I need this table. This table is important. Then blue color solution will have the yellow absorbance, then it will be measured at 560 nanometer. Greenish blue at will absorb the orange color light and will have the wavelength of the 650 nanometer. Bluish green will absorb the red light and it has wavelength of 800. So this is the filter, and the construction I have explained already two times, that when a light source of the visible range will be scattering the light, that light will be arranged by an aperture called as a slit, will be fall on a condenser which will condense the light towards the filter, and filter will be select the light of the that particular wavelength and will fall it on the cuvette, and that cuvette will have the sample, and in that analyte is going to absorb the light, and the remaining light will be passed on the photocells, which will be sensitized by the galvanometer, and galvanometer will give a reading. The colorimeter works on the Beer and Lambert law. Lambert has discussed about the thickness, or you can say the path length, and Beer has discussed about the concentration. So if the transmission is 100%, absorbance will be 0%. It is used to determine the amount of analyte present into the sample, molar absorptivity, titration, and following the progress. So this is all about the colorimeter and the centrifuge machines. Thank you very much for watching. Bye everyone. If you have any doubt, you can stay, or I'm closing the link. Okay, no doubt, no problem. So we can end the session now.